So metabolism thereafter becomes a second level. Really, this is about safety first, and everything else becomes interesting. But the safety of the elevation of ALTs is the primary endpoint.
Great, thank you.
You're very welcome.
So our third speaker is Dr. Andrew Shen. Dr. Shen is a neuroscientist in the Department of Neurotoxicology at the FDA's National Center for Toxicological Research. He has extensive experience in neurotoxicology and behavioral pharmacology. His research interests include identifying neurobehavioral consequences of developmental and adult CNS insults, including environmental contaminants, food additives, and drugs. So today he will give us a talk entitled, the title is Effects of Perinatal CBD Exposure on Developmental Outcomes, a focus on neurocognitive and motor functioning. Dr. Shen, it's all yours.
All right. Great. Can you see my screen?
Yes, I can.
Okay, perfect. All right. Well, thank you for that introduction, Jim Pong, and thank you to Norbert Kaminsky and CRIS for inviting me to give this talk today. It's going to be a little bit of a gear change from what we've just seen in the last two talks that were a little bit geared towards liver toxicology. This is more geared towards neurobehavioral toxicology, as that is my field. This is just a disclaimer statement. This presentation is not a formal dissemination of information by FDA and does not represent agency position or policy, as well as I have no conflicts of interest to declare. Just to briefly restate some of what we heard in some of the talks yesterday, in particular by Asa Waldstein, was that Americans are using CBD. What we're seeing here is a Gallup poll from last summer in 2019, June, July, that estimated approximately 14% of US adults had either tried or were using CBD products. And I think Asa kind of touched on that yesterday and said maybe Nielsen was predicting that's around 19% now have tried or are consistently using CBD products. There are a number of purported uses for cannabidiol or CBD, addiction, anxiety, depression. We've also heard a lot about anti-inflammation, potentially anti-cancer, and just general wellness. What is particularly interesting is that a number of these purported uses, like anxiety, chronic pain, and nausea, overlap with common symptoms that women experience during pregnancy. And so there is kind of a worry that women that are pregnant or might be pregnant are using CBD to treat these issues that they're having. This was an article that was published a few years ago in the High Times by Burgess Powell, asking, is it safe to use CBD products while pregnant? General take home of the article underlined at the bottom, quote, we can say that CBD use and accessibility is growing rapidly, and many women are choosing to experience pregnancy with a little help from hemp. The take home being that pregnant women may or can be using CBD products to treat things like anxiety or insomnia or GI distress. What we see in the animal research is that there's also been kind of a boom in CBD research, and we see it in the Alzheimer's disease field, in hippocampal neurogenesis, looking at the endocannabinoid system, dealing with stress, and a number of other research topics. So the preclinical and experimental literature has engaged CBD research. However, it's going to take a few years for that to catch up. As far as purified cannabidiol use, Brad and Patrick have already touched upon a lot of the Epidiolex literature. So I'm not going to go over it too much, at least a lot of the liver effects. But it is obviously approved to treat seizures associated with Dravet, Lennox-Gastaut, and tuberous sclerosis complex. There is obviously the potential for liver injury, drug-drug interactions, and reports of male reproductive toxicity in animal studies. However, in this case, the benefit of reducing seizures relative to the other risks is quite good. So it's a good product for that. The effective dose range of Epidiolex, as was mentioned, starts at 5 milligrams per kilogram per day and works up to a maintenance dose of 10 to 20 milligrams per kilogram per day. And I just wanted to show this slide to kind of give some context as to what these doses mean in terms of total CBD, purified CBD intake on a daily basis. If you use kind of a reference standard of 60 kilograms per day, you're looking at a starting dose of 300 mg per day, and at a maintenance level, 600 to 1200 mg per day of purified CBD. The take-home message here being that these Epidiolex doses are considerably higher than what a consumer might be taking. The non-clinical Epidiolex data included two studies that are particularly relevant to the neurobehavioral tox field. One was a developmental study where animals are exposed pre and postnatally to relatively high doses of purified CBD, ranging from 75 to 250 milligrams per kilogram per day. And these studies noted that there were neurobehavioral deficits that were observed at the two higher doses, 150 and 200, as well as effects on the reproductive system. So we saw things like delayed sexual maturation. In the juvenile study, where animals were exposed postnatally from days 4 to 77, what we see is that some higher doses, again, there are neurobehavioral deficits that are reported and reproductive toxicity was observed at all three of these doses, 100, 150, and 250. And as was mentioned in some of the previous talks, this reproductive toxicity has been observed in the experimental animal literature as well. Again, just for context here with the non-clinical Epidiolex dose range, that 75 milligrams per kilogram per day that was a no effect level in the developmental study is associated with plasma levels that are approximately 9 times higher than what you would get at the reference human dose of 20 mg per kg per day. In the juvenile study, there was no established no effect dose because all doses produced some level of toxicity. However, those CBD exposures were associated with levels of plasma levels approximately 30 times higher than the human reference dose of 20 milligrams per kilogram per day. The take home being that these studies are based on safety. These doses are very high relative to what will be seen in consumers, of which you would even use for consumer safety studies. As many of the other panelists have already reported, there are a number of gaps in the literature for CBD. And in particular, our group is focused on effects on the developing fetus and potentially the adolescent brain. And we're really focusing on a neurocognition and to extent neuromotor functions, as well as sex-dependent effects, reprotox, and potentially the metabolites in future studies, 7-carboxy and 7-hydroxy. The NCTR is taking a targeted approach to CBD research, given the numerous different needs of different product centers and different researchers. And in particular, we're interested if there are any behavioral effects following developmental CBD exposure, in particular on neurocognitive and motor functions, given the signals that we saw in the Epidiolex data that had to deal with the rat studies, the developmental and the juvenile studies. We're also interested in neuroimmune effects, and we heard a lot yesterday about CBD's potential effects on the immune system. In addition, reproductive toxicity. and finally, pharmacokinetics and pharmacodynamics. So we currently have underway a 2 to 3 year pre and postnatal developmental study of cannabidiol. Here we're orally dosing pregnant Sprague-Dawley rats with a purified CBD substance. It's delivered in sesame oil. This is obviously to mimic the Epidiolex formulation. Animals are exposed from GD6, gestational day 6, to the day prior to parturition. And then we directly expose the offspring using oral gavage from postnatal day 1 to 21. We're using a larger dose range than was done in the Epidiolex studies on both ends. So we're going lower and higher, using doses ranging from 15 all the way up to 350 milligrams per kilogram per day. And we're using large sample sizes. This will make any effect that we see more statistically meaningful. And in particular, like I said, we have an interest in investigating behavioral endpoints. Some of the ones that we're most interested in are, we chose because we either saw a signal in the experimental literature or there was a signal that we saw in the Epidiolex data sets. So one would be neuromotor. So this would be because we saw things dealing with spontaneous locomotor activity. So we're interested in motor learning, gait, and coordination. We also saw signals in the literature that had to deal with sensory motor effects. So we're including things like acoustic startle and pre-pulse inhibition tests that measure sensory motor gating. In addition, anxiety-related behavior, we have 4 distinct tests that we're doing in a longitudinal manner that deal with anxiety-related behavior. And a heavy focus on learning and memory, as we're doing kind of the standard Morse water maze paradigm, as well as a long operant conditioning paradigm. And the latter there, operant conditioning, we're going to use to focus in really on executive functions. And when we start talking about executive functions, we're really talking about things like behavior flexibility, the ability to attend to multiple stimuli at the same time, working memory, and time perception. And so we're going to use a number of tasks over a course of about 110 days to look at how rats, the offspring that have been exposed to CD developmentally, perform in these tasks. So this is a breakdown of the study design given in a timeline, so that you can see what type of data we're going to produce and how that will come. Animals arrive at our facility at gestational day 5, and we expose them from gestational day 6 to 22. And then direct exposure of the offspring, like I said, from postnatal day 1 to 21. It's important to note that at postnatal day 1 and at postnatal day 21, we're going to cull back the litters, and we're going to use some of the animals for histopathology and some for neurochemistry at both ages. Animals start the behavior testing at PND 21. That's going to run all the way to the end of the study at PND 180, or 6 months of age. We're going to do repeated motor function testing in a longitudinal manner, as well as repeated anxiety-related testing and repeated sensory motor gating acoustic startle testing. And over the course of roughly 110 days, we're going to do operant testing to focus in on those executive functions. The animals will be tested 4 to 6 times every week. And then at the end of this study, we'll collect numerous tissues and samples for histopathology and neurochemistry. And I think one of the things that we want to make sure we do with this study, given that it is such a large animal study that we're having large sample sizes of between 20 and 25 per sex per treatment, is to maximize the return on this study. And to do that, we're collaborating with other divisions at the NCTR, with other PIs, so that we can create supplemental studies to utilize the animals that we've already programmed for to address additional data needs. And the two most important, I think, that we identified that we wanted to see if we could add supplemental studies were immunology and reproductive toxicology. So in our first supplemental study that we're looking at, it's a Neuro-Immune study. And as we heard a lot yesterday about potential neuroimmune effects that CBD might have, or immunosuppressive effects, this is something that we really wanted to investigate. In particular, is there a potential for long-term suppression of the immune system? What might be the effects on the immune system following a developmental exposure event? And so we're collaborating with other PIs within our division for this study. And we're going to do a standard immune system challenge using LPS, or lipopolysaccharide. Same study design, but after we dose a subset of animals with LPS, we'll look at time points 2 hours after the injection and 24 hours after the injection. And we're interested in cytokine levels in blood and brain tissue. We want to look at splenocytes so we can identify and quantitate different leukocyte populations and see if there's been any change. And 24 hours post-injection, we're really interested in the effect on microglia, as they are the brain's macrophages. We're interested to see if there's a change in microglial activation state. And this is simply a timeline of that supplemental study. It's identical to the main developmental study, except that we're going to pull some subset of animals at postnatal day 21 and 180 to do these LPS challenges. We hope to get this study underway next month. The second supplemental study is a ReproTox study. And as we've kind of already discussed and other panelists have mentioned, there's existing data that suggests that higher doses of CBD have effects on, in particular on the male reproductive system. It seems reduced testicular size, delays in sexual maturation, as well as adversely affecting some other measures of healthy reproductive function, like depletion of germ cells and inhibition of spermatogenesis. So for this, we're collaborating with the Division of Biochemical Toxicology, for a reproductive tox study that fits within the confines of our larger developmental study. So we're going to pull additional tissue at already planned collection times, postnatal day 21 and postnatal day 180, for histopathology, gene expression, RNA-seq analysis, as well as measures of sperm count, morphology, and motility at the postnatal day 180 time point. And this is a timeline of that study. Again, it is an identical timeline to the main developmental study that we've already planned. We're just going to pull additional tissue at predefined time points, postnatal day 21 and postnatal day 180. And I just want to mention that in addition to these studies that branched off from our developmental study, there are several more that are ongoing at the NCTR right now. One is an in vitro evaluation of male reproductive tox that's being conducted by the Division of Genetic and Molecular Toxicology. And that is going to include, I believe, direct exposure to 7-carboxy and 7-hydroxy CBD. As well as there's a pharmacokinetic study of cannabidiol following dermal exposure in rats that's being done by the Division of Biochemical Toxicology. And one more protocol that's currently under review is another pharmacokinetic study of CBD in major metabolites, 7-carboxy and 7-hydroxy, in pregnant Sprague-Dawley rats and the pups and the offspring. That's also the Division of Biochem Tox. So I know it's a lot of information, and a lot of it is still underway. And we hope that the main developmental study will be finished in about 8 to 12 months. We hope that the Neuro-Immune study can get underway next month, and the male repro supplemental study is already underway, as well as the in vitro and the PK dermal exposure study. So with that, I have a number of people to thank, in particular in the Division of Neurotoxicology, Sherry Ferguson, our director, and the colleagues that I'm working with on this project, Tim Flanagan and Drew Gill. as well as Fong Liu and Leah Latham that will be doing a lot of the immunology for us. In the Division of Molecular and Genetic Tox, Dayton Pettibone doing the in vitro male repro study. We've also had a lot of help from the Office of Scientific Coordination from Matt Bryant and Manny Chidambaram that are doing our CBD analysis and CBD purity studies. And with that, I'll take any questions if there's time. Thank you.
Thank you, Andrew. That's a great talk. So now I think that we are reaching the last part of the session, the panelist discussion. I think all the questions can be asked and answered during this session. I will ask all three speakers to unmute themselves, and I will read the questions from the Q&A. And attendees, if you have additional questions, please submit either by Q&A or raise your hand. So here, there are a couple of questions that I got from Q&A. The first one is the FDA collaborating with Canada? How did the Health Canada already get to the point that CBD as a food ingredient is fine? Any of the speaker can weigh in.
I guess my understanding with Canada, and we've been working with a few vendors up there, is CBD in Canada from hemp is still not acceptable. It would be from considered marijuana. And they have a different way of regulating where food is a subcategory of medicine or drug, versus here we have DSHEA that handles what is supplements. And we draw that line that as soon as we go to diagnose or treat, it goes to a drug side. And so Canada's a little bit more streamlined there, but they're only doing it with CBD from marijuana. They've still not made hemp a legal product up there, is my understanding. I'm not sure that answers all the question, but that's at least most recent information we have.
I guess a follow-up that is that I think the FDA views CBD as a drug since the patent approval of the Epidiolex. Do you think Canada, do they regulate the similar way, or do you have any insight about that? Okay, maybe we can.
Yeah, I don't have a whole lot of knowledge on how Health Canada is regulating CBD use, at least in dietary supplements. I mean, but as you mentioned, we know that the real sticking point with FDA is like you mentioned, because CBD's been the subject of new drug applications. FDA categorically excludes CBD as a dietary ingredient, so they won't review it for use in dietary supplement. And I'm sure that'll be touched upon in greater detail in the next session of this, but I think I'm sure that's one major difference between the US and Canada. And again, I unfortunately am not familiar with what Health Canada is doing, but I doubt that they look at it in that exact framework that FDA does.
Great, thanks. Another question is CBD passed through breastfeeding? Dr. Shen, do you have insight on this?
Yeah, we've observed in the literature that the lactational transfer of CBD is relatively low. It's why I believe in the sponsored studies of Epidiolex, they use subcutaneous injection during the early postnatal days and then went to direct oral gavage exposure. In our studies that are ongoing right now, we are directly gavaging the pups at postnatal day 1 and just avoiding any complications that may come from unknown lactational transfer. But we've seen it to be very low.
Okay, thank you. I see a chat via the panelist. I think Sylvia may have some insight into the Health Canada. I will unmute you. Maybe you can speak out to the attendees.
Hi, thank you. So I also am not an expert in Canadian regulation, so I'll caveat that, but I have heard, I think it's now a couple of years old, that CBD isolate is regulated in Canada under a unique legislation called the Cannabis Act. And so it's not regulated the same way that Health Canada regulates nutritional health products like dietary supplements. And I pasted the exact language from Health Canada that speaks to that to help answer that question.
Thank you. There's another question for Andrew. Is the FDA focusing on addressing safety gaps for hemp-derived CBD broad-spectrum extracts, or is the focus on CBD isolate?
It's a great question. I cannot speak to what the FDA is aiming at their safety studies. What I can say is that we're most interested right now in the purified cannabidiol solutions that are related to Epidiolex, and all of the studies that I kind of outlined, our developmental study, the Neuro-Immune study, the Reproductive Toxicity study, and the in vitro study that are being conducted are all being done with a purified CBD isolate.
And Jinpeng, if I may join in, our conversations with FDA, there's definitely a concern with whether it's a full spectrum, broad spectrum, or isolate, just from a perspective that it's on the market right now. And so any and all product that's out there, they're concerned with safety. But isolating each could be areas of interest. And I don't want to speak on their behalf, but they did want to see as much as is available on the market today.
Yeah, and I think that's what makes some of the data interpretation difficult sometimes, is that, you know, we'll be comparing full spectrum products that have an array of compounds in them relative to, you know, at the bench, we're just working with CBD isolate. That's what we have, and that's what was designed for these studies. And so it does become difficult to compare, and you probably need completely separate lines of research to investigate some of these things, especially some of the drug-drug interactions that might happen if you have more than just CBD isolate, but if you also have CBG, CBN, and some of the other derivatives from that.
Right.
And I think then you look at the forms, as folks were saying before, does it get metabolized through liver or otherwise? So the consumption methodology becomes interesting. So you've got a number of variables that need to be considered over time as they look to regulate. This is not easy math.
No, and I really do think the metabolites are going to end up playing a large role here as I don't know if it was you or if it was Brad that showed the slides about the actual derivative amounts. We know that the 7-carboxy levels are going to be lower in the rodent models, and that they may not be accurately reflecting some of that exposure, but that they do reflect the parent compound exposure quite well. And so, you know, there's going to need to be probably direct exposures to 7-carboxy and 7-hydroxy in animal studies, I would think, to kind of formalize the whole picture.
Is that kind of study is ongoing, or is that planned to be done?
Some of those have already been proposed at the NCTR. I don't want to speak FDA wide. At the NCTR, we have the in vitro reproductive tox study. We'll use 7-carboxy and 7-hydroxy isolates. One of the barriers to doing that in our developmental study, which we had originally wanted to include just a 7-carboxy exposure group, is the price. Apparently, the derivation and isolation process for the metabolites is not necessarily easy or inexpensive, and it was cost prohibitive for us to include it.
So I guess a naive question is, how do they design the dosing scheme so that the compound in the biology system will mimic the CBD metabolite?
So we worked closely with colleagues at the Center for Drug Evaluation and Research to figure out at what point we get relevant plasma levels of those metabolites.
Mhm.
And that's why we did doses of 300 and 350 mg per kg per day in our animal model, as the Epidiolex data showed that the 250 mg per kg that they used approached relevant human levels, but was not quite there yet.
Okay.
And so that's why we wanted to go one step further and include these higher doses. But we also did want to range lower as we know that the Epidiolex data did not really show a defined no observable effect level. And so that's why we included lower doses as well.
Thanks.
Yeah, if I could just follow up on that. So Andrew, first of all, I'd like to commend the NCTR for really focusing also on the 7-carboxy CBD metabolite, because I think that's going to be critical to being able to then interpret those data in the context of human exposure. But there's two pieces of that. You know, one is the exposure to the metabolite, but also in the human, as that metabolite is formed very quickly, you know, the parent compound is dropping very quickly. And
Right.
so somehow, you know, trying to get the balance on that, I think it's going to be tough. And so my question is, do we know of any other animal models other than perhaps primates that might have a more similar profile of metabolism to the human than the mouse? And again, I suspect the rat's very similar, I think, to the mouse based on Brad's presentation.
Yeah, it is. I think it's probably going to be the rabbit.
Okay.
I believe in the Epidiolex studies had the most similar to humans, other than non-human primates, obviously. But that is a combination moving forward with the mouse and rat studies. They do have relatively similar CYP enzyme profiles, and so you're going to see relatively similar levels of 7-carboxy and 7-hydroxy that are produced, including even to like 6-carboxy. But
Is there any evidence that these major metabolites may be more active than the parent?
there has been speculation that 7-carboxy may be what actually drives anti-seizure activity of Epidiolex, but there are published rodent studies that have shown anti-seizure activity of the parent compound itself, so it may be a combinatorial effect, or it may just be a large species difference, that in humans the parent compound is not the most active, but it's actually the metabolite, whereas in rodents it's both the parent compound and one of the major metabolites.
And
It's definitely a complex situation.
And in terms of toxicity, is there anything around the 7-carboxy in terms of?
I don't think we know the answer to that question.
Yeah.
I don't know that that's been rigorously studied. If you're asking the question, which is it the parent or one of the metabolites that's driving liver toxicity, that's driving potential reproductive toxicity in mice, I think that's a critical data gap. I don't think we know the answer to that. We have evidence that humans are more highly exposed to the carboxy metabolite. I think I agree with Andrew that if anything, we're a little bit closer to rabbits and mice and rats than in terms of our exposure to the metabolites. But beyond that, I think that's a data gap that we still have.
Great, thank you.
Andrew and Brad, the other thing to talk to you offline is we're about to recruit for a dog study that would go rather broad. Love to delve into this issue and see if there's something we might be able to do in that type of an animal model.
Okay.
There's a question for Brad. You mentioned the data gap of whether male reproductive toxicity, which have been, where's the question just disappeared? Let me see. guess the question, somehow the question disappeared. Oh, here it is. So you mentioned the data gap of the male reproductivity toxicity, which have been shown in the mice, also occur in human. Did your literature review find the study that have shown the male reproductive tox in monkeys?
Yeah, so I think the most robust work that's been done on male reproductive toxicity in terms of, you know, the quality of the study and full disclosure and the dose used, the type of CBD used was the mouse study. That was the 34-day mouse study that was conducted and published a couple of years ago. We did come across I think there was a study in primates that was probably an older study. I'd have to go back and look at it because frankly I haven't looked at it in quite a while, but I believe we gave that a little bit lesser weight when we were looking at the weight of evidence because there wasn't critical details on the study methods missing, and we weren't able to really leverage that information to the weight of evidence. So I don't, sorry, I don't have a better answer to your question because I haven't looked at the draft in a while, but it kind of rings a bell, but I think we gave it lower weight because of study quality issues.
Thank you, Brad. I think there's a question for Patrick. Do you have any idea what range of doses that people are using to self-medicate with CBD oil? I think that referred to the study that you have mentioned.
Yeah, so we're seeing a range. It's a very, very broad range. Again, this is folks dosing or consuming on their own without any type of instruction, but a 5 milligram to over 1,000 milligrams a day is the range. Our longitudinal data that we have from our broader database demonstrates on average a little bit over 25 milligrams a day seems to be the normal consumer dose that we see. But being that we're about 500 or so about half the population has finished, the range is still 5 to a little over 1,000.
Wow.
Yeah, Patrick, if I could just follow up, kind of a similar question, or at least on the same kind of topic area. So, you know, we know that there's a tremendous amount of difference in bioavailability depending on if people have eaten recently and if they've eaten a high-fat meal and so forth. And I'm just curious, people generally that are using CBD, do we have any idea like what time of day they're using it? Do they primarily take it before they go to bed, or somehow being able to factor in the timing of when people take it in proportion to when they're actually taking their meals? Because it seems like that is a huge factor in terms of how much is actually being taken up.
Yeah, so in this study, we didn't ask for specific time, but we did ask the number of times per day and then the quantity at each dose or each consumption. So that to your point, Norbert, I think, you know, are we on an empty stomach and fasting or not might be interesting versus post-meal. I don't have the information at my disposal. We are asking journaling to questions each morning. So they're either reflecting the day prior, typically the day prior's dose, and then we catch. There is a reminder setting that allows us to catch morning and evening, but I can get back to you and see what we have on that.
Great, thank you.
Here's another question. I think it's for Andrew. What's the current status of the NCTR study? Has the postnatal testing begun?
Yes, we began our study back in geez, late July, early August of this year. So we're about a third of the way through of the live animal portion. And as I mentioned, the Neuro-Immune study, we hope to start next month. And the supplemental male repro study is ongoing as well. So they've already been collecting samples from our animals when we've called. So, yeah, so we're underway. So hopefully maybe a year from now, we'll be finished.
All right, thank you. Here's another question for Brad. In your description for uncertainty factor, there was no uncertainty factor for intraspecies variation. Please explain why this was the case. And typically there's a uncertainty factor of 10 for interspecies variation, 10 for intraspecies variation.
Sure. So yeah, we had a tenfold factor for intra-species. So we had a tenfold factor. So we in there to account for potentially sensitive people, particularly those that may have impaired liver function, and maybe other, others that may be, taking other concurrent drugs that may exacerbate toxic effects. So we did have the tenfold factor. The interspecies factor, we did not include an interspecies factor because we were deriving our acceptable daily intake level from a human study. So there was no need to account for an animal to a human extrapolation. Had we done that from an animal study, then we would have had to include that unless we had a data-derived extrapolation factor to account for toxicokinetic differences, which we don't really have complete enough TK data to do that. So that's why we had the tenfold intraspecies and the lack of an interspecies.
Thank you. I believe that's the last question from QA, but I do have an additional question for Brad. So you mentioned that you have presented this risk assessment in many of external committees and discussed with FDAs. What's the insight you can convey about the perspective of the FDA? How likely they will agree with those numbers?
So we did in January. We did visit the FDA and we presented the work we've done on CBD risk assessment to their CBD working group, which is chaired by Amy Abernathy. So we got, I think it was received well. I think they thought we had the right approach, but we didn't really get a firm answer as to whether they sort of agreed with our ADI or not. At the time, the assessment was still draft because we hadn't gotten consensus from our own health advisory board on the final ADI at that point in time. So we presented it as draft only. I think the feedback was that they agreed with our science. They liked the science. They agreed with our approach, but they didn't necessarily say they, would be fully on board with the final ADI that we had proposed. And so, yeah, I think there's general agreement, but we haven't had much follow-up with their CBD working group since then. So that's kind of where things stand there. Was there another part to that question that I missed? I think.
No, I think that's perfect.
Yeah. Okay. Thank you. Yep.
Are you aware of any other regulatory bodies or entities that propose the ADI and how those numbers look like?
I am not aware of any other risk assessment that's been done by an authoritative agency or body where they've proposed an acceptable daily intake. Mm-hmm. I am aware of there has been efforts by another, dietary supplement consortium such as Council for Responsible Nutrition that's sponsored another evaluation that we have reviewed. I'm not sure that that's in the public domain yet, but we've reviewed that and taken that into consideration. They took a little bit of a different approach that, you know, neither I nor HEB necessarily agreed with, but that's the only other risk assessment that I'm aware of. I know that other agents, I think the UK FSA has suggested an intake level that I don't know how to phrase this, that they're not concerned with at this point, but they haven't done a full or published a full risk assessment yet. And I'm sure, I don't want to steal anybody's thunder. There's presentations coming up that may speak to this, but that would be my consensus. I don't think there's been any other that I'm aware of. Agencies that have tried to do this yet.
Thank you. I think we are about the time, and this is the end of the session 3, and we'll have a break